Greetings again, Biology 101 fans. This is yet another episode from North Shore Community College's Math and Science Division, bringing you some lab videos because you're all hunkered down at home being safe, and we want to bring these to you so you can continue some lab work. Today's topic is photosynthesis. It's from Lab 8 in the Mater Laboratory Manual. We're going to be mostly doing sections 8.2 and 8.3 on different varieties of photosynthesis here. The first section is on photosynthetic pigments, but there's enough to do in the other ones to occupy your attention. So let me clue you in on photosynthesis a little bit to begin with. Almost all, not 100%, but almost all creatures on Earth require photosynthesis in their environment one way or another. There's a few microbes that can get by without photosynthesis, but photosynthesis occurs in all the green land plants, of course, but it also occurs in a lot of ocean creatures too, particularly algae and quite a few microbes. There are also microbes in fresh water that will be able to do photosynthesis, and almost everything else in those environments will depend on photosynthesis too. So, for example, animals are going to be eating either other animals, or they're going to be eating creatures that eat plants. So one way or another, we all depend on photosynthesis. It's the major source of storing energy in the biological world using light energy to make foodstuffs. So let me give you the overall reaction to get oriented, and I'll show you what we're doing for our experimental setup. It depends on light energy. If it's out inside, obviously it's going to be light energy from the sun, or it can be used artificial light too. Here today we're going to be using artificial light. We've got a lamp here we're going to be using. But it's going to take carbon dioxide from the air, or in the case of today, it's going to be from water. But it combines it with water, and then it creates or synthesizes some sort of a sugar or a carbohydrate here. This is the end product of photosynthesis. And then cells can do whatever they need to do with sugars. This is their source of energy for all other cell functions. And oxygen is made as a waste product. If you're doing photosynthesis, oxygen you just get rid of at the end. But oxygen obviously is really crucial to a lot of other creatures on Earth, like us, that breathe the oxygen, or if you're a fish in the ocean, you're going to be absorbing oxygen from the water, and that oxygen from the water can dissolve from atmospheric oxygen, but also it's produced by photosynthetic creatures in the ocean too, or freshwater habitats, whatever. So that's the overall reaction of photosynthesis. Let me show you a little bit of visible light, just for the sake of orientation too, because that's crucial to understanding photosynthesis. So here's what's called the visible light spectrum. I didn't have it written out in colors here, show you in colors. I can show you a demonstration quick here in colors, but the visible spectrum, that is the part of what's called the electromagnetic spectrum that our eyes are sensitive to, ranges from violet light, blue, green, yellow, orange, and red in that particular order. Violet and blue light is the higher energy end of this spectrum, red being the lower energy end of the spectrum. Now there's electromagnetic radiation, this is all called. If that gets lower in energy, you go into what's called the infrared spectrum of the electromagnetic spectrum, and our eyes simply aren't sensitive to infrared radiation. By the same token, energy that becomes higher than violet is called ultraviolet light, and that's also light that our eyes simply don't detect very well, so we don't see that stuff very well. But let me give you a quick demonstration of these colors. It really does occur in this particular sequence, and in white light, all of these colors are present. White light is usually the best for photosynthetic creatures because it's got all these different energies that can be absorbed. So let me give you a quick demonstration of white light being resolved, as it's described, into the different colors. So here I have a CD. This is probably an antiquated piece of technology now, and we may be in the post-CD era, but people used to use these for videos, for recorded music, and all kinds of stuff, right? In any case, this is a shiny surface. There's a series of very small reflective dots built into the surface that records information. And if I hold this against a light, if I tilt this slowly, you can see the color spectrum on the right side of that disk. And if I do it carefully, there's indigo or violet, blue, green. If I keep tilting it, then it becomes yellowish. It's hard to see the yellow. And then orangey and red. There you can see, there's the yellow, then orangey and red, and the whole sequence kind of repeats again. So the dots on this particular disk actually break up the light energy into different portions, and that's why you see different colors. So the point is, all of those colors are normally present in white light. We just don't see it with our eyes because we see the mixture. But you have to, some device like that will actually give you the resolutions of colors. So our photosynthetic creature of the day is Elodea. This is from our aquarium. It's a green plant that grows underneath the water. So this one's not going to be absorbing carbon dioxide from the air, gaseous carbon dioxide. It's going to absorb it from the water. So in this reaction, notice I've got CO2 with a little AQ next to it. AQ means it's dissolved in water. And in particular, we're going to be using a solution of sodium bicarbonate. That's here. Sodium bicarbonate is going to be the source of CO2. Right, so the Elodea is going to be sitting in this solution. It can absorb CO2 from the solution. Water's obviously in there. And it's going to be making some sugar. And then it's going to be making some oxygen gas. And the oxygen gas, again, is going to be easy to measure the quantity of this because the gas, it'll form some, not bubbles. You won't see bubbles, but it will form gas that pushes solution up. We're going to be using this tube with a stopper in it called a volumeter. And I'll construct this in a couple of minutes so you can see this. But as the oxygen is produced, it'll increase the volume in this particular tube, and then we can measure it with this pipette that's in the tube here. So the quantity of oxygen produced is a measure of how much photosynthesis has occurred, how rapidly the photosynthesis is occurring. Let me show you this tube in a little bit more detail here, and then I'll set stuff up and get it going. This is not exactly what's described in the book. There's a test tube here with a stopper, and then this is a pipette. It's a tube that's used to measure relatively small volumes of liquid. And the pipette is subdivided, so this whole pipette will contain 1 milliliter of liquid. The smallest divisions, which is what I'm going to be using to measure the displacement of liquid in here, is in hundredths of a milliliter. So not millimeters as in the textbook, as in the lab needle, but milliliters. It's just an easy way to, you need some sort of a scale to measure how much gas is being produced, and that's the way we do it. So this is going to be filled up almost to the top, little gap of air in the tube here, and then it's going to be sealed up. As oxygen is produced from the Elodea inside the tube here, it's going to produce more gas pressure up here. The gas pressure is going to push liquid up into the tube. That's how we're going to measure the progress of photosynthesis here. So what I'm going to do is I'm going to put Elodea in the tube, put in some of the bicarbonate solution, and that's going to be a volumeter. Shine some light on it, and then wait 10 minutes to see how long it takes to push liquid up into the tube. So we'll be back in a couple of minutes, and I'll be showing you construction of the Elodea in the solution and shining light on it. There is one more complication I should mention too, because we're going to have to do this measurement a couple different ways. Here is the overall reaction of cell respiration. Now, in Elodea, even though that it's a green plant and carrying out photosynthesis, it will simultaneously be carrying out cell respiration. So in cell respiration, glucose is going to be oxidized in the presence of oxygen. At least this is for complete oxidation. It's going to be making carbon dioxide, and it's going to be making liquid water. But the important point here is it's going to be consuming some of the oxygen that's being made by the photosynthesis. So these are contrary processes, notice. Photosynthesis produces oxygen. Cell respiration will be using some of that oxygen up. So what we're going to have to do is kind of subtract out, or really add back in, the quantity of oxygen consumed by cell respiration. So there is a variation of the theme. I'm going to put Elodea in the same tube, run the experiment again, but then wrap it in aluminum foil to keep it in the dark. And when it's in the dark, no photosynthesis is occurring, but it will be consuming oxygen. And as it consumes oxygen, then we can measure how much oxygen really will be consumed. We'll be able to watch that change in the volume, and the volumeter, as it's called, too. And then we combine those two things to get the net production of oxygen by photosynthesis. But to show you the construction of the volumeter with the Elodea in it, I'm first going to take some Elodea and put it into the tube here. Probably the more I put in, the better, because more Elodea means you got more photosynthesis going on, easier to measure. I'm going to stuff these little plants into the tube. Then I'm going to add the bicarbonate solution to that. That should be plenty for the plant here. You can use this to get it to the bottom. Now I'm going to add the bicarbonate solution. Almost, but not quite to the top. When I put in the stopper, this takes some practice. Notice what happens. Maybe some white background will help. But what happens is when I push that stopper in with the volume up to here, some of the liquid is pushed up into this tube. And since it's pushed up into the tube, then I can measure it against the scale. I can measure the position of that liquid against the scale. This may help you to see, maybe not, the position of that. It's pretty hard to see either way. But if you can't see it, just kind of take my word for it. It's right there against the tube. Right, so I can measure the position of that thing with the scale on the pipette, and that's why we're using a pipette. Now what happens is, since there's this little bit of gas up in top here, as the oxygen is produced, that's increasing the pressure in this gas, and that's what's going to be pushing the liquid up the tube as oxygen's being produced by photosynthesis. So it's going to be sensitive to temperature because gases are going to be expanding as heat rises. So I'm going to put this into the test tube now, and I don't want to put my hands on it again because my hands will actually have enough heat on them to actually increase that temperature. The other piece of the apparatus setup here is going to be this beaker full of water. This is a heat sink. This is an LED light, and it really won't get too hot. But just to be safe, we still have this water here between the test tube with the Elodea and the light, because the water will be able to absorb some of that some of that heat from the light, and any of the heat comes off the light. So I'm going to turn on the light, and I'm going to wait until this starts to rise. It's a fairly bright light. This may be detrimental to the effect of the video. The camera might adjust. But what I'm going to do is I'm going to wait for this to start rising, and then I'm going to record positions here. But before I do that, let me clue you in again on the setup of this particular tube here. So in this tube, we've got a white light, no block to the light at all. That's going to change later. And we're not using any filter. It's just a white light coming through the water. In this case, we've got the Elodea sprigs and sodium bicarbonate solution. And I'm going to record again the position at time 0. That's when I choose when this thing is moving well enough And then 10 minutes later, I'm going to come back and record the position again. So I'll show you this when I record the time at 0 and when I record the time in 10 minutes. So I'm going to turn the light on, wait around for it to start rising, and then I'll show you that, record this. 10 minutes later, I'm going to record it again. Well, we've waited a couple of minutes now, and the photosynthesis is definitely kicking in. So I've seen this thing, position of the liquid move here. I'm going to record it now. It's going to be at a position of 0.0. It's going to be 0.32 milliliters. That's the position at 0 minutes. I'm going to start a timer. In 10 minutes, I'm going to come back and read it again. I'm not sure if I can capture this on camera. This is going to be a difficult one. But I can actually see bubbles coming up in that test tube. So let me see if I can show you that, just so you're convincing, just so I can be convincing that there's actually a bubble coming up here. Not right. There was one that went right then. I'll wait until one more comes up. But it's coming up. They're coming up off the tips of these Elodea. Occasionally there's bubbles coming up here. Let me see if I can. There's one. Bubbles of oxygen coming off the Elodea plant. So we're going to come back in a few minutes. After 10 minutes, it's already been a minute and a half elapsed. So we're back. 10 minutes is up. It's been moving kind of slowly, but it has absolutely been moving. In fact, I'm going to call it after 10 minutes. I'm going to add in a digit here. I'm going to call the position here 0.29. Let me get a little look. I'm going to call it 2. I'm going to add in an extra digit on the measurement here. And I can do that because when I first read this, I waited until the liquid was right on 320. These are called significant figures. It's not trivial. All right, so I'm going to subtract these for the total movement. So the total movement is 0.320 milliliters. Mine is 0.292 milliliters. So it's going to be 820. So 0.028 milliliters is the total quantity of oxygen that we recorded as being produced in here. And you can see the bubbles coming up. Again, I'm going to be summarizing this on a separate document for you, and I'll be posting that for both you and your instructors. The next variation is going to be using some aluminum foil. So what I'm going to do, we won't need the light for this either. It's going to take this tube and wrap the whole thing in aluminum foil. Aluminum foil is opaque to light. It won't let any light in there. So the Elodea will be sitting in the dark. If it's in the dark, it can't do photosynthesis. If it can't do photosynthesis, it won't be producing oxygen. And if it's not producing oxygen, any difference in the oxygen level that I can record here is going to be due only to cell respiration, oxygen consumption by cell respiration. So for this particular setup, what I've done with that aluminum foil, there's no light because there's an aluminum foil blocker on it. We're not using a filter, it's just not getting any light at all. It's still the same Elodea, still in the same 3% sodium bicarbonate solution, but I'm going to do the same thing. I'm going to record, this is a variation on the procedure from the book. I'm doing the same procedure as the other one, just because it's going to be easier to do the arithmetic. And I kind of doubt it's going to be much variability. In any case, I'm going to record the position, what I'm going to call time 0, record the position of that liquid in the tube there at 10 minutes again, take the difference, and that's going to be the oxygen consumed. If the oxygen is going to be consumed by the plant when it's in the dark here, if this oxygen is going to be consumed, then we will see this volume here decrease a little bit. So you can see that you'll be able to see the tube, the liquid recede in that tube, if it moves at all. But we'll see. So I'm going to give it a couple of minutes to let it sort of like get used to the new temperature, because I just held it in my hand. And then I'm going to come back, record it, position at time 0, 10 minutes later, record the position again, and that will be the amount of oxygen consumed by cell respiration. So it looks like things are starting to reverse here. So I'm going to record the position of the liquid again in this tube. And then I'm going to write it down here, 10 minutes, we'll be back. So if I hold the paper in the back of this, I'm going to call it 0.284. I'm eyeballing this last digit because I've been used to doing this. Right, so start a timer. So the position here is 0.284. And again, this is in milliliters. 10 minutes later, I'll come back, record the next position. And that's going to be, again, the amount of volume of oxygen that's being consumed. So we're back again after 10 minutes sitting in the dark. At least the Elodea has been sitting in the dark. And I'm going to read it has moved a little bit, but not very much. I'm going to read this as 0.286. Sort of in the middle, so very little movement really. So it's not a whole lot of oxygen consumption going on, but it's worth noting anyway. So again, this position is 0.286 milliliters. So if I do the subtraction, 0.286 milliliters minus 0.284, I'm sort of eyeballing that third digit. But it's significant. So 0.02 milliliters is oxygen consumption, as opposed to oxygen production from the photosynthesis. So the next variation on this lab setup is going to be using a green filter. Here's a piece of, probably see this better against the white background, a piece of green cellophane. I'm going to take the tube with the Elodea in it, get rid of the foil, and this time I'm going to wrap it in this green cellophane. So the green cellophane is going to be a light filter. That means that any light that the Elodea is going to be exposed to is going to be green rather than the white light. So why do we want to do green? The point is the photosynthesis is not sensitive uniformly to all the different colors of light. Plants look green. Why do they look green? Let me get this taped. Okay, so let me show you the light spectrum again. Here's the light spectrum again. So you have to really realize that plants look green because of the photosynthetic pigments in plants. So if it's appearing green, it means it's not absorbing green light. If the light's absorbed, you're not going to see it. So chlorophyll is the pigment, there's several different kinds of chlorophylls, but chlorophylls are the pigments in green plants. There's a couple of different varieties of those things. But since they're appearing green, it means it's not absorbing green light. There are other pigments in photosynthetic creatures that will absorb different varieties of light or have different appearances. So some of them are carotenes, and you all know what carotenes are because you've probably eaten orange carrots and orange sweet potatoes. The orange color of those vegetables is due to carotenes. Carotenes tend to look yellowish and orangey. There's also pigments called xanthophylls, and those tend to be yellowish colored. In any case, all those different pigments allow photosynthetic creatures to absorb more energy from this visible spectrum. But because there's a lot of green light that's being reflected, you can probably expect that green light is not being absorbed. So what I'm going to do is do something kind of crude here. This you can call a color wheel. And what I'm going to do is break it up into halves. This is a crude way of estimating what colors are being absorbed from the color that's appearing. So here's R for red, orange, yellow, green, blue, and violet. So it goes like this. You look at the opposites. So violet is opposite of yellow, green is opposite of red, blue is opposite of orange. If something is appearing green, it's because it's principally absorbing red light. If something appears blue, it's because it's absorbing orange light and so forth. It's kind of crude, but it's only a rough estimate, but it works in some cases. And with the chlorophyll, they're fairly complicated compounds. They too tend to absorb in the red region. They also absorb in the violet region too, violet into the blue region too. So there's a big peak, and if you look in your lab manual, there's an absorbance spectrum of chlorophylls there, and some of the xanthophylls. But there's a big peak of absorbance from the chlorophylls in the violet and blue region. There's also a peak in the reddish region. But again, it does not absorb green light because the green light is principally reflected. So the point is this: what if we expose the photosynthetic creature here, the Elodea, only to green light? So only green light is going to be getting through this particular filter. What happens if we give it only green light? What would happen to the photosynthetic process? So we're going to do the same thing. Same kind of setup. I'm going to turn the light on. Light shines onto the tube here. And I'm going to be measuring the position. This time there should be photosynthesis occurring. I'm going to be measuring the position of this fluid in the pipette here. Let it sit for 10 minutes, record it again. So this particular setup, let me just give you the complete description here. This particular setup looks like this. We've got a third version of this. So we're going to use white light again. We're not blocking the light with the aluminum foil from last time, but we are using the green filter. So that's the new variation, and which is why I underlined it. Green filter here. Same Elodea sprigs, same 3% sodium bicarbonate solution. I'm going to be measuring the position of that liquid in the pipette at 0 minutes, measure it again at 10 minutes, and we'll look at the difference and see how much oxidant has accumulated in that 10 minutes. So I'm going to switch on the light now. This will probably blow out the camera for a few seconds. The camera adjusts. And again, I'm going to record the position of the liquid in this tube. It hasn't moved yet. I'm going to wait a couple of minutes, see if it moves, come back. Or actually what I'm going to do is wait for it to get to a convenient measuring point, and then I'm going to measure it 10 minutes later, measure it again. So it looks like this stuff is starting to move a little bit. So I'm going to record the position. It's at a relatively convenient place to record. So I'm going to record this as the initial position of 0.280 volts. It's sitting right on one of those division lines. So that's why I'm using it now. And it will probably go up a little bit, but we'll see in 10 minutes. Start a timer, be back in 10 minutes. So it's just been 10 minutes. I'm going to record the position of this liquid again in this tube here, if I look at it, it's moved a bit. I'm going to call this 0.258. I'm going to record it here. 0.258 milliliters. We'll do the subtraction. 0.280 milliliters to begin with, minus 0.258 milliliters. So this is 220. So 0.22 milliliters of oxygen. This is oxygen produced, or at least that's the gas that's ended up being produced. So again, I have this all summarized for you on a separate document. There's one more variation we're going to do. We're going to be looking at carbon dioxide uptake rather than oxygen consumption. Here we're going to be looking at carbon dioxide consumption versus oxygen production. So I have to tell you about particular properties of carbon dioxide. And we're going to be using something called an acid-base indicator. So we're going to be playing around with carbon dioxide this time, as I mentioned. Carbon dioxide really does have acidic properties when dissolved in water. It will decrease pH if you dissolve some in water. And we're going to be using what's called an acid-base indicator to monitor that. I have here a dye called phenol red. It's a dye that changes color. So it's, because it changes color with pH, it's called an acid-base indicator. If the pH is less than 6.8, it's going to be yellow. If the pH goes above 8.2, it turns red, or what's called fuchsia, to be the official term. It really is the official term for the color here. I'm not making that up. Somewhere between 6.8 and 8.2, it's reddish of different shades, kind of orangey. But it will respond to carbon dioxide being dissolved in water. So let me just give you a demonstration of how this stuff changes color first with acids and bases, and then I'll show you with some CO2. So I've just got some tap water in this beaker. I'm going to be putting that in these two tubes here. And I'm using tap water rather than distilled water, because distilled water, when it's exposed to air, it absorbs some CO2, and then that can actually have a relatively low pH. So if I use tap water, it will be more likely to be closer to pH 7. So tap water in here. I'm going to give each tube a drop of phenol red. Maybe 2. Maybe 3. That should be red enough. Okay, so if you look at the white background here, this tube now looks red because of the red dye in it. Phenol red. Let me give it a mix. Pinkish red. Pinkish red in here. So what I'm going to do is just to show you the color change first, is I'm going to add some acid and base, just so you don't think I'm making it up. So here's some acid, 0.5 molar hydrochloric acid. I won't need much. Fairly dilute acid, but I'm only going to put a drop in. See what happens. And you can see just with one drop of that acid, it's turning yellowy already. Right, so not that much acid, you get a nice yellow color instead of red. Low pHs, under pH 6.8, it's going to be that yellowish color. And again, above 6, it will be more red. So here is some base. Here's some dilute sodium hydroxide. This will be a base. I'm going to put it in this tube, and we'll see what happens to the color. And again, I'm only going to put a drop in. Now, it's red to begin with, I know, but if you can see that shade changes a little bit. It gets more of a fuchsia red, right? That's the description. This is no longer just red. It is fuchsia. But it does change shades. Now just to show you what happens with carbon dioxide, here's some tap water. And there's a little drop of dye in there already, but I'm going to put a couple of drops of dye in. Make sure it's red enough. So I'm going to add carbon dioxide to this. Where am I going to get the carbon dioxide, you ask? We all have carbon dioxide, right? We're breathing it out. So all I have to do is breathe on top of this thing. So if I blow my breath on this stuff, it'll change a little bit to yellow. It'll take a few breaths. Oh, changing maybe a little bit in shade. A little bit more orangey now. You can see the color changing here a little bit. Orangey more on top, reddish on the bottom, and that's just because the CO2 that's being absorbed into the water from my breath. It's almost yellowy now. So what happens here is that little bit of CO2 is changing that to yellow, right? So what's going to go on with the plant? Let me give you the reaction of photosynthesis again, just to remind you. Here is the reaction of photosynthesis. So what I'm going to do, I'm going to use this dye to monitor carbon dioxide uptake or consumption. So here's CO2. It's the reactant in this reaction room. So what I'm going to do, I'm going to put the same Elodea in this tube, but it's not going to be a bicarbonate solution. It's going to be some tap water along with this red dye. I'm going to start it out as yellow, and the CO2, as it's being consumed from the yellow solution, let's see what happens when the CO2 is consumed. Acid-base indicator, really monitoring how much CO2 is in solution. So I'm going to add tap water to this, some dye, breathe on it, and then put the Elodea in. Tap water. I'm not going to fill it entirely full yet. So I got to be able to mix this stuff. Put a little bit of dye in here. Just barely make it red. Get a glove so I can mix it. Maybe another drop of dye will be good. That's relatively pink. Now I'm going to breathe on it. Change it, see if I can get it to be yellow. I bet you never knew you're breathing out acid, breathing out CO2. But we are. CO2 really does have properties of an acid. It gets kind of complicated when you're dealing with it in water, but carbon dioxide itself is an acid. It's turning kind of orange now. Maybe that's good. I'll take that. So I'm going to do is fill it with more water. Hopefully it won't turn back to pink. And I must put the I'm going to put the Elodea back in. The Elodea has been hanging out here waiting to do its stuff again. So no bicarbonate this time, just carbon dioxide from the air in my breath. Stick this back in, and I'm going to let this sit in light and photosynthesize. And then I'm going to time it. So right now, it's still kind of yellowish. All right. I'm going to turn on the light, let it start photosynthesizing. As it's photosynthesizing, CO2 will be consumed. As the CO2 is consumed, we will be watching for a color change. Putting on the timer to see how long it takes for the color to change in that solution. Let me lower the camera so you get a better look at it. So there it is at time 0, relatively yellowish. Does this work? Kind of. Well, let's see what happens with the color in a few minutes. So things have been photosynthesizing away here. It's been like 9.5 minutes. I'm going to shut the light off so you can actually see this a little bit better. Then I'm going to start the stopwatch at 9:45, almost. So if you could look at this now, here's the Elodea again with the phenol red in it. See how well you can see that in the camera, but there's a distinct pinky color up here in the top where the Elodea is. And it's still a little bit orangey at the bottom, but it's certainly not yellow like it was before. A little bit orangey in the bottom, a little bit pinkish up on top here. And again, pinkish because the CO2 has been consumed by the plant. And if the CO2 is being consumed, the pH would be going up. And if you notice this, the fluid in the tube here is slightly pink too. I just put a little bit of the dye in there. It could have used more dye, but then dye could be toxic to the Elodea too. In any case, there's a little bit of that pink color. We're going to call it 9 minutes, 45 seconds. So here is the setup that we used for this one. White light, no block, no filter. Elodea sprigs this time in tap water and phenol red. That's what's giving it the yellowish color. And again, for the time for the color change, so it's going to change to pink near the Elodea. And it took 9 minutes, 45 seconds to change that pinkish color. It was gradually changing. I could have waited another minute. I could have done it a minute earlier. But I just wanted to have enough color there to convince you that it really was pinkish. That's it for photosynthesis for this episode.